Respected organizers and my dear friends, I am Dr. ARUNA Chakrawarti, Associate Professor of Zoology, Government Dungar College, Bikaner. My topic is therapeutic potential of aloe vera against radiation and cadmium-induced changes in the brain, kidney, and liver of Swiss albino mice. Aloe vera is the gift of nature. It is a miracle being used medicinally for centuries. It belongs to the family Liliaceae, botanically known as Aloe barbadensis. The medicinal properties of various part, leaf, aloe gel are rich in minerals, magnesium, calcium, chromium, copper, iron, etc. Enzymes, amino acids, and vitamins as antioxidant. Oxidative stress in cells and tissue results from the increased generation of reactive oxygen species. Decrease in antioxidant defense potential. Antioxidant defense system represent a complex network with interaction, synergy, and specific task for a given antioxidant. The plant contains substantial amount of antioxidant including vitamin E, vitamin C, flavonoid, carotenoid, and tannins. In recent years, the study related to the interaction of living system with ionizing radiation has focused on physiochemical or biochemical events associated with the cell membrane. Radiation is beneficial for therapeutic purposes, but high doses or continuous exposure can cause different syndromes. Ionizing radiation causes formation of free radicals, which are potentially dangerous to the cell and its constituent. The reaction of hydroxyl and peroxyl free radicals on biomolecules is important in the field of physiology and pathology. Cadmium is an industrial and environmental pollutant arising primarily from battery, electroplating, pigment, plastic, and fertilizer industries. Cadmium is dangerous because humans consume both plant and animals that absorb cadmium efficiently and concentrate it within their tissues. Cadmium shows various mechanisms of toxicity in particular species under different experimental conditions. It has been demonstrated that cadmium stimulates free radical production, resulting in oxidative deterioration of lipid, protein, and DNA, and initiating various pathological conditions in humans and animals. Once absorbed, cadmium is rapidly cleared from the blood and concentrated in various tissues. Chronic exposure to inorganic cadmium results in accumulation of the metal mainly in the liver and kidney as well as in other tissues and organs, causing many metabolic and histological changes, membrane damage, altered gene expression, and apoptosis. Aloe barbadensis, commonly known as Guarpata, the most important medicinal plant in the Indian traditional system of medicine, the Ayurveda. Several parts of the plant are used to treat the variety of diseases, but the most important is the leaf. Herbal plants are potential source of phytochemicals of pharmaceutical interest, act as antioxidant, anti-carcinogenic, and antimicrobial preclinical studies. Experimental studies have reported that aloe and some of its phytochemicals also exhibit radiomodulatory, chemoprotective, free radical scavenging, and immunomodulatory activities. Having these unique properties, aloe vera could be used as protector against radiation and cadmium. Therefore, the present study is planned to evaluate protective efficacy of aloe vera against radiation and cadmium-induced changes in the Swiss albino mice. The adult healthy Swiss albino mice were procured and maintained at Animal House of Government Tungar College, Bikaner, and provided balanced mice feed and water ad libitum. The animals were divided into 7 groups to investigate protective effect of aloe vera in mice, brain, kidney, liver against radiation and cadmium chloride alone and in combination also. The experimental animals also provided oral doses of aloe vera. A minimum of 5 animals from each group were sacrificed by cervical dislocation and autopsy at each post-treatment interval of 1st, 2nd, 4th, 7th, 14 and 28 days. Liver, kidney and brain was taken out and was kept at -20 degrees Celsius. The biochemical estimation of total proteins, glycogen, cholesterol, acid and alkaline phosphatase activity, DNA and RNA was done in laboratory. The animals were divided randomly into 7 groups, 4 mice in each group. The first group received distilled water and served as control. Group second animals were administered orally with cadmium chloride. Group third animal were exposed by 6 gray gamma radiation. Group fourth animal treated with cadmium chloride and radiation. Another set of animal, group 5, 6 and 7 were orally administered aloe vera with cadmium with radiation and combined treatment of radiation and cadmium chloride. In all the drug-treated animals, the aloe vera was given 7 days prior to radiation, cadmium or combined treatment group. Now radiation sickness. Animal exposed to 6-ray gamma radiation exhibited sign and symptom of radiation sickness. These mice were found as lethargic and weak. Food and water consumption was reduced, although general activities of such animals were apparently normal during all 28 days post-irradiation. Animals pre-treated with aloe extract and later exposed to radiation did not show any sign and symptoms of radiation sickness. A significant weight gain in this animal was observed as compared to control with normal food or water consumption. In group 3rd and 4th, the animal which were irradiated with or without cadmium chloride, the value of protein content of mouse kidney and liver showed an increasing trend, and in brain decreasing trend up to day 14 in non-drug treated group and day 7 in aloe treated group, that is 6 and 7. Thereafter the content decrease and increase up to day 28. On the contrary, the animals of group 2nd and 5th treated with only cadmium chloride with or without aloe vera, the value of total protein declined in kidney and liver. This observation indicated that the amount of total protein is adversely affected by the cadmium chloride. The change is less severe in aloe-treated group, showing protection provided by drug. A significant increase in the number of ribosomes may occur due to their increased mobilization from endoplasmic reticulum, and this leads to the increased protein synthesis. In case of brain, it is believed that heavy metal cause the death of ribosome by acting on membranes and also the cellular death, thus decreasing protein synthesis. The value of glycogen content increased in all the tissue up to day 14 in non-drug treated group and day 7 in drug treated as compared to normal. Thereafter, the value declined up to last autopsy interval. After combined treatment, synergistic changes were seen and earlier and faster recovery was noted in aloe-treated groups. An increase in the value of liver glycogen in rats with 450 rat exposure for the first 6 days was observed, which might be due to glyconeogenesis. Decrease in the value of cholesterol content was observed in liver and kidney of mouse, while in brain the value increased up to day 14 in non-drug treated group and day 7 in drug treated group. In cadmium treated group, value of cholesterol declined due to stress response caused by irradiation to stimulate synthesis of steroid hormone, increase ACTH secretion by pituitary, leading to decreased cholesterol concentration. The decrease in cholesterol level in present study may be because of radiation-induced free radical that can damage the glial cell, leading to depletion of cholesterol level. An elevation in the value of acid phosphatase activity was reported in Swiss albino mice. The increased acid phosphatase level seems to be characteristics of tissue damage by radiation. Cadmium damage to lysosome by membrane lipid peroxidation that causes release of hydrolytic enzymes like nucleases and acid phosphatase, which may account for an increase in the enzyme activity. Another possible reason for elevation of acid phosphatase activity may be the damage of liver after cadmium intoxication. An increase in the alkaline phosphatase activity was observed up to day 14 in non-drug treated group and day 7 in Aloe treated group. The efficacy of aloe vera in modifying acute cytotoxicity of cadmium in male rats has also been evaluated. The results suggested cytoprotective potential of aloe vera in acute cadmium toxicity, which could be due to its multiple role in biological system. The concentration of RNA increased on day 1 and continued so up to day 14 in non-drug treated animals and day 7 in aloe treated animals. Thereafter, it declined on day 28. This increase in cellular RNA may be due to an increase in RNA concentration of the surviving cells. Ability of DNA to transcribe RNA is not affected quantitatively, but the length of the chain of RNA molecule reduces. Increased gonadotropin secretion after irradiation has been reported. This increased gonadotropin secretion may accelerate RNA synthesis after higher doses of radiation. Decrease in DNA content was noted during the experiment. DNA is the critical target of radiation damage in living cell, which may lead alteration in the functional state of cell and future of cell death. Damage caused to DNA molecule by irradiation leads to other metabolic alteration also. Ionizing radiation induced damage to cellular DNA, which is of prime biological significance. The type of damage include strand breaks, base damage, elimination of bases, and sugar damage. It has been shown that the exogenous application of aloe vera increases glutathione level in the tissue on one hand and maintain acid group and increase protein synthesis on other hand. Hence, antioxidant as mediators of disease prevention. Man is exposed to number of toxic substances in the environment, including radiation as well as toxic metabolites and reactive oxygen species generated within the body. Results obtained from the present study indicate that the natural medicine found in aloe vera, including antioxidant and other phytonutrients, substantially protect the living tissue from radiation damages. However, further research is needed, especially regarding the mechanistic aspects of this protection. Radiation damage to tissue causes degenerative changes, and combined treatment with cadmium shows synergistic effect. Aloe treatment of mice prevent oxidative damage, depicted superoxide scavenging, and depicted peroxidation inhibition. So, aloe vera may be used for developing novel radioprotective drugs. Thank you.